Yeah. So, good evening, everyone. Let me introduce myself to you people first. So, I guess I'm audible to you all, right? Yeah. Yeah. Good evening. So, basically, I am Srijan Sen. I am a research scholar of Indian Institute of Technology, Kharagpur, and I am doing my research basically on protein engineering and structural biology. So, I would be taking the live session on the course of experimental biochemistry, which is being taught by Professor Sagota Das Gupta and Professor Shomode of IIT Kharagpur. So basically, this course deals with a lot of basics on the experimental techniques that we use in our daily labs. Those who are dealing in research, or whether you do it in BTech or MTech or in your bachelor's. Okay, so it will deal with lots of techniques. So we need to know the theory. We need to understand how it goes, and then we can discuss a lot of things about it. So this course, we will start with this course with the basics of the understanding. Then we move with the different purification techniques. We will be going through a lot of understanding on how we purify the proteins, what proteins basically are. We will talk about nucleic acids and its purification, and then we would be seeing about the detection of these nucleic acids as well as the proteins via the different immunoassay techniques and all. So welcome you all once again to this particular course on experimental biochemistry, and I hope to see a good number of attendance in this live sessions, because these sessions are very important for a proper understanding of what you are seeing in the lectures, as well as I would be discussing a few question and answers in my in the later half, after I go through a summary of what you have been taught in the week 1. And the question answers, what I do is basically it is a quiz type where I put forward you the questions which can be of previous years, which can be collected from the different sections of the different examinations, and then it is open to you all to give an answer, and then I talk about it and discuss the answers to you all. And if this makes good sense, we can start with today's lecture. If you have any questions in the beginning about this particular course in a general basis, you can ask me. I would give a minute, and then in the meantime, I'm sharing my slides with you all, so we can start it soon. And additionally, this live session will be taken on Friday. I would be taking it on Fridays from 6 p.m. to 8 p.m. So yes, join this particular NPTEL course. Ask your friends, colleagues to join this one. It's a very good course, and you can learn a lot from it. when you hear the lectures, you attend the live sessions, and you watch the practical classes. It's going to be a great experience for all of you, whether you are in a bachelor's degree or a master's or even a scholar doing research and all. Okay. Okay, so let me share my slides with you all. Yeah. So I guess I'm visible to you all, right? Just give me a confirmation. Yeah, great. So let us start. Fantastic. Okay. So what we'll be talking, what we will be talking today is about basically the concepts that will be covered is firstly, I would be discussing about accuracy and precision on a very general basis. So I guess all of you know it, but I would be just touching it and trying to solve a few question and answers such that you have even a better understanding. Then mostly we would be speaking about the concepts of water, ions, acids and bases, pH, the Henderson-Hasselbalch equation, and buffers. So let us start with the accuracy and precision, which are the two sides of the coin when we talk about measurement. So these are the very important factors when we are talking of any kind of measurement, because these are very, very critical when we talk about anything. So what does precision basically mean? So precision is basically the closeness of two or more measurements to each other. So it is basically, it does not depend on whether your system is accurate or your measurement is accurate or not. It's basically how close your two or more measurements are. So it can be separated. Now, precision can be categorized into repeatability and reproducibility. So these two terms mean the variation that arises from the conditions when these particular conditions are kept identical and repeated, and which are taken for a very short period of time. You called it repeatability. And reproducibility is a factor that you are repeating the experiment over time and time for a longer period, and yet you are getting the same measurement. So repeatability is when you're doing it for a very short period of time, and reproducibility is when you are doing a particular experiment. Maybe you do it in the month of January and you do it in the month of March, and again in the month of December, you get the same result. So it's over a longer duration, and you call it reproducibility. And this reproducibility is a very important term when we talk of biology, okay? Because reproducible experiments or reproducible data is very important factor in all sorts of research basically. Accuracy, on the other hand, speaks of the ability of the instrument to measure it correctly. Okay, so for that, what we need is we need to know the value, the prior value, because the value has to be known from before, such that we can tell whether my measurement is accurate or not. For that, a prior knowledge is important. But for precision, you do not need a prior knowledge. It's about the closeness. You do an experiment today, you do an experiment tomorrow. It tells whether it's precise or not. But accurate, you need to have a prior literature value from which you are going to relate. So when we see, so let us see what if a measurement is accurate, and whether it has to be actually very precise. The answer can be understood when we draw a graph versus accuracy versus precision. See, we have like a wagon wheel, okay, and we see this. These are. Let us imagine that we have a dart over here, and then someone is throwing the pins at it, and we see the first image that shows that it is a very scattered kind of a plot, we what we say. So it is a very scattered distribution, which we call accurate but not precise. So how we can call it accurate? Because the mean value of it, of all these collected distributions, bring it to a mean which is close to the center. So we can call it accurate, but it's not precise because the measurements are not very close apart. What about this? It is not even close to the center, and it has a very scattered towards the boundary, whose mean is not even close to the center, and we call this distribution not even accurate nor precise. What if all the darts were thrown at one particular place? So maybe this can be a sort of an error which we call. So it is not close to the center, but all the distributions are coming over here. So an idea on accuracy or precision need not only tell about the data. Rather, if you get a plot like this, what if you need to normalize it and bring it to the center? So it can also tell you about whether your instrument has any kind of error or not. That is why the measurement is very, very important. Now this is precise as well as accurate because the mean value is very close to the center, as well as the dots are also very close to one another. Okay, so if you look at this particular example which tells the volume of a liquid is 20.4 ml, so which of these measurements represents a value with good accuracy? So I guess you all can answer this. So give me an answer, please. All of you, what would be the best answer for this particular question? So the volume is 20.4 ml. So there are four options given, and which of the following options represents a value with good accuracy? Let me see. Oh, sorry. Yeah. Option D. Okay. Why do we call it? Yes, because the mean value of all, we need to calculate the mean over here. Yes, accuracy, we can understand if the mean is very close to the accurate or the known value. Fantastic. So D is the correct option. So I will just mark it over here. Yes, because the mean of the DA comes to about 20.4 ml. For the others, it is not. Yes, absolutely. D is the correct option. Okay, let's move on then. So so just the difference between accuracy and precision. So accuracy is the closeness of with the true value with the quantity that we are being measured, whereas precision speaks about the reproducibility. And measurement can be accurate, not necessarily be precise, but your measurements can be precise but not necessarily be accurate, and that talks about the error of a particular instrument. And accuracy can be determined with a single measurement, whereas precision needs several measurements, because for anything you have to take a multiple measurement, because reproducible data is what we want to see as well. But accuracy you can understand if you are getting an accurate value because you have a prior knowledge, but for an instrument to be precise you have to do it multiple times to get a precise value, if not an accurate one. And accuracy speaks of the degree of conformity, whereas precision speaks of reproducibility. So the correctness of the data and the repeatability of the data, or the reproducibility of the data. So this is another example which says if the weather temperature reaches 20 degrees and it is 20 degree outside, so we can call it accurate. But if the thermometer continuously registers the same temperature for several days, and that is 20 degrees, we also call it precise. But if the thermometer is not registering the same temperature, maybe it's registering about 25 degrees, then we call the error of the thermometer or the error of an instrument. Now, for example, if you take the measurement of the mass of a body of 50 kgs and you get something values like this 47.4 and etc, etc, your weighing scale is basically precise but not accurate. And if you get a scale which gives you a value of a varied distribution, it is more accurate, but it's not very precise. So we want an instrument that has to be very precise as well as accurate. Otherwise, we get an error in the data, which is not good for our experiments. Putting forward another question or an example, you can speak where you have a new 5 rupee coin that weighs about 6 grams. Okay, and you have two weighing machines that gives different weights. Okay, so it was measured 5 times and you get different weights. So which of the statement would you say it to be correct? Okay, whether A is accurate or B is precise, or B is precise, A is accurate, or what? So another question for you all. I guess it's an easy one. I guess you all can do it, but still, just a minute. Right. Okay, I got an answer. Great. B. Okay, so for accuracy, what you are calculating is the standard mean. Okay, so you are calculating the mean of a particular weighing machine or the values registered. How can you talk about the precision? What do you measure for a precision? When you are calculating precision, what do you actually need to calculate? Can anyone say it to me? So for accuracy, we calculate the standard mean. For precision, what do we measure? Or how can you tell it is being precise? Sometimes can be a huge set of data and then you cannot do it manually. Then what you're going to calculate to talk about the precision? Closeness to the true value? No. That talks about, no. How can you check a huge set of data? That you can do manually. You can check it manually and tell like it is very close. For you have 5 data. What you have a big data set of 5,000 or 10,000, you cannot tell. You cannot go back and check all the data. For that you need to calculate something. What is it? It's very easy. You're calculating the standard mean. How do you calculate an error? No, mean will give you the accuracy of an instrument. Mean does not give you the closeness. What gives you the closeness? So the closeness means it's a difference. So there is something. Think about it. Probably we'll get an answer like the standard deviation can give you an idea on the precision. Okay, just think about this particular precision question. Okay, anyways, let's move ahead. Okay, so B is the correct answer actually. Fine. So now let's move. So this is about accuracy and precision. Now we will talk a little bit about acids and bases. So we all know what acids basically are, what bases basically are. So this is an idea of taking an example of a water molecule where a free hydrogen ion does not actually exist in water. Its attraction to the water molecules makes it so strong that H plus exists as a hydronium ion or the H3O plus ion. So water is such an important molecule which can act as both an acid as well as a base. So water acting as a base, it happens when it is responsible in abstracting a proton, and when it acts as an acid, what it does, it donates a proton. When do we see it? When we are adding HCl to water, it acts as a base. Water acts as a base, whereas when you're adding water to a basic substance or a basic solution, you see to be water acting as an acid, and there is an exchange reaction taking place between the two reactant molecules. So the concept of conjugate acid-base pair came from the Bronsted-Lowry theory, where this particular conjugate pair consists of molecules or ions that are related by the loss of one hydrogen ion by an acid and the gain of one H plus by a base. So every acid-base reaction that is taking place contains two conjugate acid-base pairs because of an H plus that is transferred in both the forward and the reverse directions. So if you see the two examples where you have the the hydrogen fluoride, which acts as an acid that loses one H plus to form its conjugate base, which is your fluoride, and water acts as a base by gaining one hydrogen H plus ion to form its conjugate acid, that is the hydronium ions. Whereas if we add water to an ammonia solution, you get the NH4 plus and the OH minus, where we have the conjugate acid base pair as NH3, which acts as the base, and NH4 plus, which is your conjugate acid. And on the other hand, you have the H2O plus, which is the base, which is an acid, and it loses the H plus to form its conjugate uh, base, which is the OH minus ions. Just a minute. Right. Fine. So we can understand this conjugate acid base pair theory more better when we look into the dissociation of water molecules. This is disturbing. Okay. Right. Fine. Okay. So when we see that water is basically amphoteric in nature, that means it can act as both an acid as well as a base. So what we see over here is that we have two water molecules, where one water molecule acts as the acid, having its conjugate base, and the other water molecule is the base, acting as is the acid, and it has its conjugate base, and the other one is a base where it has its conjugate acid. So in pure water, there is a forward reaction between the two water molecules that transfers the H plus from one water molecule to the other. So one water molecule acts as an acid and the other molecule acts as a base. Now, this is very important when we want to derive the dissociation expression of this particular water molecule. So this is in short, and the equilibrium constant of which is represented as K over here is indicated by the concentration of the hydronium ions and the hydroxide ions divided by the concentration of the water molecules. Now we consider that the constant concentration of pure water that we have over here, we can eliminate the lower portion, and therefore we can write it as equilibrium constant for the water dissociation expression. So the water dissociation expression, or Kw, is indicated by the uh, concentration of hydronium ions multiplied to the concentration of the hydroxide ions. Now, when we have, so it has been determined previously, okay, like when these experiments were done in at 25 degrees, and the concentration of uh, okay. So, like we have the when these experiments were done at 25 degrees. So for each of the, so for pure water, the concentration of the hydroxide ions and the hydronium ions were similar, and that is 10 to the power minus 7 molar. From this, at 25 degrees, Kw is equal to 10 to the power minus 14 molar square, you can tell. And that is the equilibrium constant value, or the water dissociation expression at 25 degrees. Now, why this equation is very important? Because if we know the concentration of the hydronium ions of a solution, and we want to use this to understand the hydroxide ion concentration, or vice versa, we can use this equation in a similar manner, and we can get the value of the hydroxide ion concentration or the hydronium ion concentration, considering Kw value will be 10 to the power minus 14. So we do these values as in terms of molarity. So molar, so when these values are given millimolar or nanomolar, we need to convert it to molarity and bring it to the same value. You all know this, know all of these things about, and then we get an idea about the concentration. So why do we need this? Because the concentration of the hydronium ions or the hydroxide ions will give you an idea about the pH. No one speaks about like the concentration of HCl is something like when you go to a lab and do an experiment, you ask what is the pH. You ask the pH of this particular buffer. You ask the pH of a solution. You do not go and ask what is the concentration of the hydrogen ions or the hydronium ions do you have over here. And that is why we have brought the scientists have brought it to a normalized scale, or we can call it as the pH scale. And when this concentration or the pH scale, we know about it, it gives you an idea about the acidity, the basicity, or the neutrality of a particular solution. When the pH is less than 7, it is an acidic solution. When it's a neutral solution, it means that the pH is close to 7. And when it's a basic solution, it is greater than 7. So the pH scale gives you an idea of the different types of solutions that you are having. Now from the pH of this, you can also get an idea about the hydronium ions or the hydroxide ion concentrations. You can get about it. These are the different regular day things that we either consume or we use in our daily lives. Like we have no one consumes an HCl solution, definitely. We use it for the different cleaning purposes. We have the lemon juices in the category of acidic solutions. You have tomato, apple juice, orange that are acidic in nature. Whereas neutral, we have drinking water, we have blood plasma, which is 7.4, and in the basic solutions, we have the milk of magnesia, we have NaOH solution, bleach, ammonia, etc., so on and so forth. Okay, so for in a pH scale, what we also need to understand whether the solution is acidic or basic, we use indicators. Okay, now these indicators can be of many types depending on the range of pH that it can detect. Like if we want to detect a solution that has a pH range of say 5 to 7, we are going to choose something like a methyl red or a bromothymol blue, and depending on the color at the lower pH or the higher pH, we can tell whether the particular solution is acidic in nature or basic in nature. So these are the commonly used indicators, and the pH ranges of these indicators are mentioned over here. You can take the help of it to understand about you can use this to determine the what do you call the acidity or the basicity of the particular solution, because it changes, it has a different color at a lower pH and it changes color when it goes to a higher pH range. So what is the pH and pOH of solution? So we speak very well the pH scale, the pOH scale and all. Okay, so what these things basically are. Right. So pH of a solution is, okay, so the pH scale is the logarithmic scale that corresponds to the concentration of hydronium ions or the H plus ions in an aqueous solution. If we think about it in a mathematical term, it is the negative logarithm to the base 10 of the concentration of hydronium ions. For example, if we have a lemon juice that has the concentration of H3O plus ions to be 10 to the power minus 2 molar, that indicates that it has a pH of 2. And we know it by this manner that pH 2 means it's an acidic solution. However, the concentration of the hydronium ions would be determined, would be very important when we want to put this particular value in the different calculations. Similarly, the pOH scale also is the negative logarithm to the base 10 of the hydroxide ions of a solution, and pOH is minus log of hydroxide ion concentration. However, the pH and pOH concentration, the multiplication of these two concentration gives you the water dissociation constant value, or the Kw, that is 10 to the power minus 14. So 10 minus log to the base 10 of 10 to the power minus 14 is basically 14. So the addition or the summation of the pH and the pOH will always be 14, and that is the general theory, the general you can tell the general uh, equation that you need to keep in mind. pOH plus pOH is equal to 14, because when you're given the pH value or the pOH value, you will also get an idea about the other factor which is not mentioned over here. So I'm not going into this example. It's a very easy example where you have a pH of 3.5 and you want to know about the pOH. So you just subtract it from 14 and you get 10.5. That's the pOH. And then you might be asked to find the concentration of the hydroxide ion, and that you can get about it when you do the antilog of the pOH value. So in summary, what we got to know is that pH is the negative logarithm of the hydronium ions, and pOH is minus log of hydroxide ions, where H plus and OH minus are in molarity, and your hydronium ions and the hydroxide ions multiplied gives you 10 to the power minus 14 molar square. The summation of pH and pOH is equal to 14. An acid is defined as a proton donor, a base is a proton acceptor, and AH, what is mentioned over here, is an acid that donates a proton, and A minus is your conjugate base, whereas on the other hand, B plus a B, which accepts a proton, acts as a base, where your hydroxide minus is your conjugate base and your BH plus is the conjugate acid. Now, so this is about pH and pOH, but acids, bases can be of different types. You can have weak acids, strong acids, weak bases, and strong bases. And how do you define them? So let us first understand the weak acids and about the pKa. The other things are fall in on a similar note. Strong acids dissociate entirely, it's our consideration. Strong bases dissociate, so strong acids and bases have a dissociation power that reaches to a completion, so it dissociates completely, so we do not need to worry about it. But when we are talking of an equilibrium constant, equilibrium, where it talks about a middle point or a point that we do not have an idea about. So equilibrium means a point where you want to reach such that there is no exchange between the two different sides, and it reaches to a boundary condition. That is where the concept of weak acids and weak bases come. So what are weak acids? So the strength of an acid can be determined by its dissociation constant. The acids that do not dissociate completely, or the bases that do not dissociate completely, are known as weak acids and bases. Right, so the dissociation of an acid can be expressed as HA, which is in like reversible arrow. You have the H plus and the A minus, and your dissociation constant Ka is equal to the H plus ion concentration multiplied by the A minus ion concentration. That is the multiplication of the products, the concentration of the products divided by the concentration of the reactant. When your Ka value is less than 1, that is your Ha or the reactant concentration is more than the product concentration. That indicates that your Ha is not significantly dissociated. That is, your Ha is a weak acid when your dissociation constant is less than 1. So at Ka less than 1, it is Ha behaves as a weak acid. Similar to pH, the value of the Ka can also be represented as pKa, which is equal to minus log of Ka. So the larger the pKa value, the weaker will be the acid. So pKa is the constant for each conjugate acid and its conjugate base pair. And most biological compounds are weak acids or weak bases. And from here, from this part is where we get an idea about buffers, which I will be speaking about. Now, acids can be also polyprotic in nature. That is a substance that is capable of donating more than one proton. Okay, so it has numerous protons that is exchangeable with the other molecules. We call it a polyprotic acid. It can be a diprotic or a triprotic if it has a capability to donate 2 or 3 protons respectively. So it will have multiple dissociation constants which we can name as Ka1, Ka2, Ka3 and so on and so forth, depending on the number of times it basically dissociates. So some examples of polyprotic acids which we use in our daily life is the in different experiments and uh, in our labs is the sulfuric acid, that is a diprotic polyprotic acid, and it has two equivalence points of its capability to donate two protons. We have the sulfurous acid, that is also a diprotic acid. We have the orthophosphoric acid, that is triprotic in nature, and we have the carbonic acid, that is also diprotic in nature. However, I need to mention one thing over here is that the it does not mean the number of hydrogens it has, because this is a very common question that is asked numerous times, where a particular molecule is given and it has 4 hydrogen atoms. It does not mean it is a tetraprotic polyprotic acid. 4 hydrogens, if they are exchangeable, if they dissociate, if this molecule is capable of donating all the 4 hydrogen atoms, only then we can call it a tetraprotic acid. Else the number of hydrogen atoms it is capable of donating, only that it will be its polyprotic nature, or what we call it as the polyprotic acids, depending on how many hydrogen atoms it's actually donating. So we need to keep this thing in mind. Okay, for that we need to know the structure of the particular molecule and check if the particular hydrogen is exchangeable between the two molecules. Okay, so the pH meter, what it does is basically measures the pH of the solution. That is what not I am worried about. It does measure the pH of a solution. What I want to send a message is that the components that it has, so it has a pH measuring electrode that is dipped in the particular solution for measuring the pH of the particular solution. You have a reference electrode that normalizes the pH, and it has a reference electrode which you use for cancelling you need a blank solution, okay, for using it as a reference to whatever the solution you are using. Otherwise, how will it know? So like you have 3 solutions, you know you have a pH of 4, you have a pH of 7, and you have a pH of 10, and these are the reference solutions, and you use this electrode to measure these solutions. You calibrate the instrument and then you use it. You have a temperature sensor and an input meter over here that will tell you the pH of the particular solution. So the pH measuring electrode is a glass electrode that uses a thin glass membrane that is responsive to its changes in hydrogen activity, which is why it measures the pH changes. It works by a precision system where we have a voltmeter in its probe that measures the difference between the voltages of the two electrodes that we have, the measuring electrode and the reference electrode, and this meter translates into a pH value which you see displayed on this particular screen. So these components are very important, and what is the role of this particular instrument, what's the principle behind it, is very important. So those are the two important points that I have mentioned over here, which you need to keep on in mind. Like, no one is going to ask you what is a pH meter used for, but people, there might be questions like, what is the pH, what are the components there in a pH meter, what is the principle behind the working of a pH meter. So in even in this lecture or in my lectures, what I would be focusing on is not the entire of what lecture has been said to you. I would be focusing on the important points, what you need to keep in mind, and just keep these things in mind, and that will help you crack any exam, not only this NPTEL exam, but any exam on this particular topic. Okay, so yes, what are the things that you need to keep in mind? The points to remember is that there are a few factors that leads to the improper pH measurement. So what if the glass electrode, there is a storing solution for the storage of the electrode. So if the glass electrode is not stored properly, it can lead to improper measurements. If the glass electrode is old, there is a rust on it, or a coating of salts and other samples, like the previous person who has used it has not cleaned it properly and has kept it, and there is salt deposition, rust deposition. Yes, there are a lot of solutions you dip this electrode into, so there can be a lot of things. So if there is a coating or a clogging on this diaphragm medium, then you will not get proper measurements. So what is it important? The first thing that you need to do is properly clean the probe or the electrode that you are dipping it into a solution. It will help you in two ways. First is that it will give you proper measurements. Second, it will avoid the cross contamination of the sample that you are using. So always do this. It's highly recommended. Number two is that you always need to standardize or calibrate your pH meter. How do you do this? For calibration, we have some common known pH of a few solutions. So you take these few solutions, 3 or 4 solutions, you calibrate it, you know the value of that particular solution, you know the pH value, you enter it over there. So the machine knows, the instrument knows that this is the solution that has a pH 4. This is the solution that has a pH 7. So now if there is a fluctuation in the concentration of the salts or the H plus or the OH minus ions, it can easily detect. So calibration is very important for an analytical instrument, and we do that using our reference page. So when we do that, also after using it, we need to clean it, we need to give some time for the instrument to cool down, and then we stop the instrument, keep it in a good condition for the better run of these instruments. Now, this is a experimental biochemistry course. We need to have biology whenever we are speaking of something. So we have spoken about accuracy, precision, instruments, water. When does this pH come into play into biology? That's very important. So biological molecules, blood, which has a pH of 7.4, that is also having a pH, and many enzymes function in that particular pH, and that's a very fantastic, you can call it beauty of nature, because of which we have that particular pH in which many enzymes are highly active inside our body, as well as the temperature that we have. Okay, so the biological molecules are very sensitive to pH because they have moieties, which includes amino acids, nucleic acids, which includes your nitrogenous bases, the hydrogen bonds, the sugar phosphate backbone, which are very sensitive to changes with the environment in terms of changing the acidity or the basicity of your body or the different biological substances that you are using. So so the pH basically affects the solubility of these biological substances and the structural and the functional activity of the enzymes, so basically enzymes are your proteins, that's a combination of the different amino acids. So affecting the acidity of this particular affecting the pH of this particular solution or the body can lead to loss of structure as well as function. Therefore, it is very important part in biology. pH plays a very important role. Now, now we are talking of pH and we do not speak of buffer, that's not possible. So buffers, so biological systems use buffers. So we use to make proteins, we make enzymes, we do a lot of experiments, we do activity assays. For everything we need is a buffer that maintains a pH. So what is a buffer? We all know what buffers are. You add an acid, you add a base, you add something to it, it does not change vastly. Like in a water molecule, you are adding HCl. Okay, there is a very sharp change, or you add ammonia solution to it or NaOH solution to it. There is a very sharp change. What buffer it does, it basically abstracts that extent of change and it restricts the change. So so what we think is like a buffer is a solution that resists a significant change in pH upon the addition of small amount of acid or a base. I mention small amount because you can say that if I am adding in a liter of solution, I'm adding about 100 ml or 200 ml of buffer of an acidic solution to it, then it is not like that it will never change. It has a limit, it has a capacity. Buffer does not have a, it's not like that buffers will have a very high capacity to resist the change. Everyone's have a resistance power, we have, buffers have, so everything has life. We can think about like think it in that manner as well. So it is a solution that resists a significant change when we are adding a small amount of an acid or a base to it. Like when you see this, there is a sharp change when you are adding an acid or base, but here in a buffer, it changes small. Fine. Now, chemically, chemically it has a very high significance. Why? Because a buffer is a solution that can be formed when we are mixing uh, when we have a mixture of a weak acid and a weak base. What do I mean by a weak acid and a conjugate base? I have explained before. I am putting an example forward that will help you to understand. Now, so if we are preparing a bicarbonate buffer, that is a mixture of the carbonic acid, which is the weak acid over here, and you have the bicarbonate ion, that is the conjugate base over here. So the combination of these two salts, or you can call these two ions, will be very critical in the formation of a bicarbonate buffer of a certain pH. Now all OH and H plus ions added to a buffer are consumed and the overall hydronium ion concentration or the pH is not altered. So you can see the equation over here. So for any weak acid conjugate base pair, the buffering range is its pKa, which has a plus minus 1. So this is not very much important. What we need to keep in mind over here is that it's a mixture of a weak acid and the conjugate base. Now, this is a very important equation from where lots of questions will be asked. There will be lot of numericals that will be there in your exams and in many other exams. So the Henderson-Hasselbalch equation or the HHE. So dissociation of a weak acid can be mathematically described by this particular HH equation or the Henderson-Hasselbalch equation. So we have HA that dissociates into H plus and A minus. So we can keep this equation in our mind as we move ahead with the calculation. So it has a particular dissociation constant, that is our Ka. So Ka can be represented as the concentration of the products divided by the concentration of the reactants. So if we write it in this manner and we go with the logarithm on both the sides, what we get is the log Ka, which is equal to your log H plus concentration plus we put log into this entire thing over here. And when we put the negative sign on both the, or we exchange these two values, like we put it over here and we get over here, what we have is This minus log of H plus is our pH, minus log of Ka is our pKa. So pH is equal to pKa plus log of conjugate base by weak acid. Okay. Now, so if we indicate Cb as the conjugate base and we have Ha as the weak acid, we can write this equation in this manner, and this is the equation that we have to keep in our mind. Okay. So another time I'm repeating, pH is equal to pKa plus log of the conjugate base, or the or you have the A minus divided by the weak acid or the acid. Okay, so this is the Henderson-Hasselbalch equation. When you have in a certain condition, when you have the conjugate base that is equal to the concentration of the weak acid, means an equilibrium condition has been reached, and that is where pH is equal to your pKa value. So the applications of Henderson-Hasselbalch's equation gives you an idea about the ratio of the conjugate acids by to the weak acid. If the pH is known, you can get you can calculate the pH if the concentration of the conjugate base and the weak acid is known. Definitely the pK value has to be known as well. You can calculate the pH of the weak acid solution of a known concentration. Determining the pK value is also possible if you bring the concentration of conjugate base and conjugate acid and measure its pH, because under that situation pH is equal to pKa. We can also calculate the changes in pH when a strong base is added to a solution of weak acid, and this can be represented in our titration curve. And finally, we can calculate the PI, or the isoelectric point. Now, there are many buffers that we use in daily experiments in biological systems and all. Phosphate buffer has a very high buffering capacity and it's highly soluble in water, and therefore this buffer is the most widely used buffer in the biological systems. However, there are a few disadvantages which this phosphate buffer might have. So it inhibits some enzymatic procedures and enzymatic reactions. It precipitates in ethanol conditions, so the phosphate buffers cannot be used to precipitate DNA or RNA. And there is a possibility of its interaction with magnesium and calcium ions, and does interfere with many biological reactions as well, in which cases we need to use a different kind of a buffer. But in most cases it does work very well because of its high buffering capacity. Like if you want to do an experiment at different pH of say 5 6 like 4.5 7 6 and 8 and like that, if you have a very high buffering range, you can use this phosphate buffer. So buffering capacity, this is a very new term, and we need to know the definition of this particular term. Is buffering capacity is its ability of the buffer to retain its pH when strong acids or bases are added in a medium. It's a very general definition when we talk about buffers as well. So it's similar to a buffering capacity. So buffer capacity corresponds to the amount of H plus or OH minus ions that is required to neutralize the buffer. Another very commonly used buffer is your tris buffer. So tris buffer is also highly soluble in water. It's physiologically inert in nature. However, its pK is around 8.1, so it has a low buffering capacity. That is, you can use it only at a range of 6.8 to 8.8. Okay, so it's a physiological range, but you can go as low as 6.8, okay, and go as high as about 8.2, 8.8. Okay, and it is temperature sensitive, so it may react with normal pH electrodes. So special electrodes are required. So this is about the tris buffer. Now, just a minute. Uh, So another kind of buffer which we know about is the zwitterionic buffer. So zwitterionic buffer it was introduced by a scientist named Good. So we called it as the Goods buffer. So these are the different Goods buffer. I have listed it. It's not very important, but maybe there might be some question like um, which one of it is not a goods buffer or something. Okay, so just for your exam, for your um, maybe one or two questions, so I have put it as like that. It's not very important, but you just need to keep it in mind. Okay, so a few examples includes a mess that for pH 6 you have the mops at pH 7, you have tris pH 8, you have taps at pH 9, and the chest at pH 10. Okay, and there are other goods buffer as well that have a different buffering capacity and buffer range, and you can use it if you want to use a Zwitterionic kind of buffer, but this is just for your information. Now, there are different methods for the preparation of buffers. We can directly put it in a Henderson-Hasselbalch equation. We know the pKa of a particular solution or a particular okay, we know the pK of a particular solution, and we add only that much amount of salt to bring it to a particular concentration of the conjugate base and the amount of acid that we are adding. And directly by putting it into the Henderson-Hasselbalch equation, calculating first we calculate it the specific ratio of the salt to the acid, and then we add it at the desired amount of water, we get the actual pH. That is that can be an easy way to do it. The next one is that we do it using the titration method, another very commonly used method where we have a highly acidic or a highly basic solution, and then we add drop by drop amount of acid or base to it to bring it to a buffering range or a pH, a desired pH that we want. And the third, which is you can call it a shortcut technique or a technique that might be there, might not be there, that is a prior literature value or a prior literature knowledge, where there are published tables, you have a pH of that, you know how much amount of what to add. And there are so many ways, like for example, if you want to prepare a phosphate buffer, you need not do it in that manner. Okay, there are so many tools and websites which you just put the amount of solution that you want to make, the concentration of the phosphate that you want to keep, and the pH that you want to keep, and it will give you all the information of how much amount of dibasic, monobasic acid, phosphoric acid that you want to add. And these are the different ways of how you can calculate the buffer, you can prepare the buffer. Okay, so the titration method, it's the most simplest of the method. So you have the two components, an acid and a base, you titrate one against the other. So it's a very easy understanding. What you do also, you can add an indicator to this particular solution, and you have an acid over here, you add the base drop by drop, and the moment the color changes, it comes to the desired pH that you want. So these were the titration experiments that we did in our chemistry classes. Most of you, I guess, have probably done titrations, so it's a very easy understanding. But the disadvantage is that it might require a lot of salt or the base or the acid, depending on whatever the pH you want to do. And it's relatively slow, and the variations in pH may not be considered, right? Because it is not giving you an exact value to the particular pH. Rather, if the color change, you have to add an indicator, and the change in color will give you an approximate value. Therefore, the titration method is not very accurate. Okay, the other techniques I think I have discussed about, so we need not worry about all of that. So these are the ways of how you can why buffers are important, what role it has in the biological system, and the methods of preparing these buffers, calculating the pH of the buffers, and then concept of acids and bases. I think I have covered the most of how much is I wanted to actually. Okay, I would like to shift into the question and answer part, but I would give you just 2 minutes to ask if you have any questions. If you have any questions, I would be happy to answer. Then we can move to the question answer part. Yeah, I guess. Yeah, it's being recorded, so yeah, fine. Any questions? Anyways, if you have any questions, maybe you want to. Anyways, I think the NPTEL team will be sharing with you the recorded lecture, which I would be also uploading in YouTube, and also they would be sharing with you the slides. So fine, if you have any questions, you can just put me a mail. Okay, I will just drop my email ID at the end. Just give me a reminder. I will just put drop my email, so you can just put a mail over there if you have any questions with the theory, or even later on when you go on to solve your um, like question, you have the assignments and all. So if you, even if you have any questions, you can just drop me the question, put a screenshot of the question and just drop me. I will be happy to answer you back in mail as well, so it's fine. Okay, because I'm absolutely very open. Feel free to reach me out. You don't have to worry about. And I'm very prompt in very. I will be promptly answering you as well if I'm not very busy. So it's fine. You can just be very open with your queries and all. Fine. So let's move with the questions. Or you need a 5 minutes break? You need a break, or should I go with the questions? Break not required. Fine. Yeah. Okay. So let's move with the first one then. So I will put you the questions and you would be answering me. So most of these questions, so these lectures are the ones from where you will get direct questions. You don't have to worry much about it, um, like what is going to be there and what not. So it's fine. There will be direct questions only. I guess you can answer all of this. Okay, so let's start. I am waiting for the answers from you all only. And I will just say which is the correct answer. Okay, so we have 18, 17 students as of now. Good. Yeah. So many of you have enrolled. Ask your colleagues to just join for the live sessions. Yeah, so all of you are saying C? Yes. So I just discussed about it. So C is the correct answer, right? I'll just bold it. Yeah, right. So C is the correct answer. Okay, the next one. It's another direct one. The concentration of H plus ion in 1 millimolar of NaOH. So you have to find the concentration of the H plus in 1 millimolar of NaOH. D. C. Okay. Now we are getting to our different types of answers. Okay. Good. D. So how do you calculate it? Let's try to solve it. It's easy though, but fine. Um, the text box. Right. Sorry, just wait. So we have been given the concentration of NaOH. So 10 to the power minus 3, that is molar, right? So 10 to the power minus 3 molar is the hydroxide concentration. Now I have been asked the concentration of the H plus ion. Okay, because so if it is an H plus ion, the simple equation is 10 to the power minus 14 divided by I have 10 to the power minus 3. Fine, because I have to calculate the H plus ion concentration, and when you calculate this you get the value which is D, not C. Okay, right. So D is the correct answer. Just for those who are telling the option C, for them I just put it over there. Fine, right. So I guess D is clear. Fine, next one. Right, if pH is equal to pKa in Henderson-Hasselbalch's equation, then what is the which of the following options are correct? I just mentioned when pH is equal to pKa. What happens over here is pH is equal to pKa plus log of conjugate base by conjugate acid. So in a condition where pH is equal to pKa, which of the following option will be correct? Only one answer. No one else? Yeah, A is the correct answer, and I've just said it in few slides back, that the conjugate base will be equal to the conjugate acid concentration when pH is equal to pKa, and we can use this even in determining the pKa of certain solutions of whom we are not aware of. If we are adding the equal concentration of base and the acid in that particular solution and we calculate the pH, so just keep that in mind. Okay, Next one is we need to do some calculations. Okay, so we have to calculate the pKa of lactic acid given that when the concentration of lactic acid is zero point 0.1 millimolar and the concentration of lactate is 0.07 molar. Okay, so the concentration of lactic acid is 0.01 molar and the concentration of lactate is 0.07 molar. The pH is 5. So what will be the pKa? So you people try to do. Let me also try to figure it out. How to what can be done, and tell me an answer. C. Okay. So what we have equals to pK plus log of CD by A. Okay, then what we do, we don't know the pK. A is equal to 5 minus log of and what is the value? Minus something is like 0.84, right? Is equals to 4.16. Yeah, fantastic. So C. Yeah, good. Good. So C is the correct option. I guess every one of you have given it correctly. Yeah, good. Great. So C is the correct option. Okay, next one. Ah, okay. What would be the effective pH range for MOPS buffer? This is something that you have to keep in mind. Okay. So what is the effective pH range of MOPS buffer? So let us see where we spoke of MOPS buffer. Okay, so MOPS buffer have a pH of 7, and if we check over here for MOPS, right, so it has a pH of 6.5 to 7.9. Okay, yeah, so option B will be correct. So there can be a few questions like this. Okay, so for that you need to only know about maybe the mean will give you an idea about what can be the range of it. Like in none of this, the mean is like around 7. So if you know the pH of this particular buffer is around 7 or 8, you can tell in which range the effective pH range of this particular buffer. So for as we know that 7 is for the MOPS buffer, so we are choosing a range of 6.5 to 7.9. And remembering that, we'll just do it. Okay, the next one. Which of the following is a zwitterionic buffer? Zwitterionic buffer, goods buffer. Which of the following is goods buffer or a zwitterionic buffer? Yeah. So this is also a direct question. I will not go into details of this. It's the Tris buffer. Fantastic. Okay, the next one. Okay, now a few numericals again. Grab a calculator if you have near you. So what happens now is there is a researcher who accidentally puts 1.6 gram of NaOH in 1 liter 0.5 molar acetate buffer of pH 7.47. Now, what should be the change in pH considering the pKa is 1.8 into 10 to the power minus 5 for acetic acid? So, what would be the change in pH? So we are looking for the change in pH, not the pH. Try to do this numerical. How will you do it? So I have seen one person giving an answer. Can you tell me how you are doing this? pH. That is what, yeah, we know that. But how you're going to calculate the pH over here? That is what I'm trying to find out. Yes, B is the correct option. That is fine. That is absolutely fine. Not a big deal. But how are you going to calculate the pH over here? You have been given in grams. So what you're going to do over here is, you know, the molecular weight of NaOH. You're going to first find the concentration, right? And then what you're going to do? How you're going to place this equation? So you know pH equals to pKa minus log of conjugate base by acid, but you do not know. You have mistakenly added 1.6 grams of NaOH. How are you going to do that? Mass by molar ratio into volume. Okay, and then that you did. But how are you going to place the equation? So you have a pH, you have a pKa. Sorry, you have a pKa. You do not have the pH. So first you find the pK. That's fine. Then you have zero point 0.5 molar of acetate. So how you go like that? So you take an x. So you have 0.5 minus x divided by x, and then you try to find out x. Or how do you do it? It's actually very difficult to write it over here. Otherwise, I could have shown it. Just Just give me a minute. Just a minute. Right. Yeah. So if you can see over here, how is it actually done? Yes, B is the correct option, that's fine. But what you see over here is that first you take the molecular weight of NaOH. So that's 1 moles. Now 1.6 grams of NaOH, you calculate it in number of moles, and then what you do is, like for HA or whatever you're taking, you have acetic acid over here. So you write this particular equation, you know that this is zero 0.5 molar, so this side is also going to be 0.5 molar. But what you have done in the middle is that you have added a little bit of NaOH in the middle. So you have calculated in the terms of moles. Okay, so that is about 0.04 molar, and you have, now you put it like, so this is 0.46 moles and 0.04 moles, and here you have 0.54. How you are getting it is like, it's like zero 0.5 minus x, and here it's zero 0.5 plus x when it is in the equilibrium condition. Okay, so see the change. So minus x over here and plus x. So the minus x over here is 0.04 moles. So now the new concentration of your becomes is 0.46 moles minus x and 0.54 moles plus x. The hydrogen ions does not matter. So you will have over here it's plus x only, because the new concentration is over here, and now you need to get the pH. But for the pH you need to also know about its change in the molarity or the change in the concentration when the NaOH has been added. So that is not known to you, so the change is considered over here as minus x and plus x on the product side. Now when you put it in the equation, you get the Ka. So you have, so okay, so you know the Ka, and from there you get the value of x. From the value of x you put it in the pH value, the equation, and you get it as 4.82. Now you subtract it from the previous value, which is 4.74 or something, whatever is given over there, and then you get 0.08 as the value. So this is a bit of a complicated, but once you get an idea of how it is actually done, it will be very, very easy. I guess I'm clear. Okay, if not, just ask me once, I will explain once again. Am I clear to all of you? Yeah, great. Okay, so I guess we can move with the next question then. Fine. So if Ka value for a weak acid is given, and you have to find the pKb value of a solution. So what will be it? So you are given the Ka value for a weak acid, and you have to find the pKb. So what will be the pKb? How will you do it? So we can calculate the pKa, right? pKa is equal to minus log of Ka, which is equals to something that is given, which accounts for 5.25. Okay, then what is pKb? How will you get pKb? So it's similar. pK plus is equal to 14, right? So then you subtract it, just it's easy. Yeah, fantastic. Yeah, that is the right way. So how much you're getting? 8.75. Yeah, that is fine. So B is the correct option. Good. Okay. The next one. Why does a buffer solution resist any change in pH? Why does a buffer solution resist any change in pH? They give unionized acid or base on reaction with acid and base. They have reduced H plus OH minus ions. Acids and bases in the solution are protected from attack by other ions. They have a fixed pH value. Yeah. Only a few people are answering. I want answers from all. You don't have to worry whether it is wrong or right. Just answer, because try answering it, because I would be discussing it anyways. So just put up an answer. Think and just give an answer. There are many people who are not answering. All of you have to answer actually. It will help you. Right, A is the correct option. They give unionized. Yeah. See, you people know the answer. You people can answer. So don't just wait. Just put an answer. Yes, that's the correct answer. They give unionized acid or base with the um, on reaction with the reaction. So you get the buffer has resistance power. They do not have the reduced H plus or OH minus ions. That's absolutely wrong. They do not have a fixed value, and they are not even protected from the other ions. So that's also incorrect. So they have unionized acids or base on reaction with the acid or a base. Simple. Fine. The next question, which there are four compounds and the pKa of the four compounds are given. So which of the following statements is or are true? Acid A dissociates to a greater extent in water than acid B. C and D are basic in nature, whereas A and B are acidic in nature. D is conjugate base of C. Acid A is more likely to be polyprotic than acid B. Which of them is or are correct? There can be one or more than one answer correct. Think a little bit from the perspective of a strong acid and a weak acid, and from the polyprotic nature as well, and definitely the acidity and the basicity. I don't need to speak much. So try to answer from. Okay, good. Yeah, I'm waiting for more answers. So A, okay. So let us first think about what the pK value is actually telling. So when the pK value is lower, it means the compound or the solution is more acidic in nature. That is, it can be called a strong acid. If the pK is very high, we can call it a strong base. Similarly, if the pK value lies somewhat between 5 to say 8 or 9, we can call it either a weak acid if it is on from between from 5 to 7, or if it's between from 7 to 9, we can call it a weak base. So if we just keep this particular thing only in mind, let us see the options first. The first one, acid A dissociates to a greater extent than acid B, which means acid A is a stronger acid than acid B. That is what it is trying to ask. So yes, because acid A has a lower pKa than acid B. So option A is absolutely correct. Okay. Next. The next option is C and D are basic in nature. Yes, they are indeed basic in nature, whereas A and B are acidic in nature. Yes, so this option is also correct. Okay, the next one, D is conjugate base of C. No, we cannot tell that because we do not have any information on it. So C is wrong. And acid A is likely to be polyprotic in nature. That is also, we do not have any idea on it. So A and B are the only two options which are correct. C and D we can see it's basic, A and B are acidic, but the first option is very tricky because it stronger acids dissociates more compared to weaker acids. So A and B, those who have given the answers of A and B are correct. Okay, the next one, buffers used for biological experiments should have the following characteristics. So what are the following characteristics that buffers should have in biological experiments? A, 1 and 2 are correct. Okay, let's wait a few more minutes. Chemically stable and water soluble, that is correct. So 1 is correct. Should not interact with biological molecules. That is also correct. Should have a specific pKa. You cannot tell this very surely because it should have a specific pH. Having a specific pKa may or may not be the correct option over here. And it should not have a higher ionic strength. It may or may not have, it depends on the type of experiments. The ionic strength is not very critical over here. But option 3 is a very tricky one because specific pKa does not mean the solution is going to have a specific pH. Therefore, option 3 is not correct. Many people would be putting the right on 1, 2, 3 and giving an option of D. But yes, option A is only the correct option because a specific pKa does not mean the specific pH. Only under certain condition pH is equal to pKa. So option 3 is not entirely correct. Okay, so only option A is the correct, that is 1 and 2 are the correct options. Okay, buffers with high buffering capacity means, what does buffers with high capacity mean? High buffering capacity mean high pH value, high pKa value, higher concentration of salt, higher concentration of acid or base present. D. Okay. Okay. Yeah. D is the correct option. Its higher concentration of acid or base is required to change the pH. That is the right answer. D is the right answer. Now another question we have, a buffer is prepared from 65 ml of 0.2 molar NaOH and 35 ml of 0.2 molar NH4Cl. What is the pH of the buffer solution? and pk and kb is given. Kb of NH4OH is given. This question can also be done on a similar manner like the researcher question, where the person added NaOH, 1.6 grams of NaOH. Okay, this can be done in a similar manner. So just you people do the calculations and just let me know the answer. So first you have to calculate the initial molarity, and then you have to calculate the change. Like in the reactant side you have that particular molarity that you are getting minus x, and on the product side you have plus x. Then you have to calculate x and you have to find the pH. But in this case you will get POH because the pKb value is given. Okay, so keep that in mind. Here pKa is not there, so you have the pKb value. Anyways, I guess you people can do it. Okay, just first find the initial concentration of the reactant. The reactant is basically your NH3 plus H2O. You write the equation, you write NH4OH, and you write OH minus, and then whatever you want to do, you put over there. You find the concentration of NH3, you find the concentration of the NH4 plus ions, and then you calculate the change, and ultimately you will get the POH value as 4.48. I'm just writing over here. Just wait. So your POH will be 4.48, and okay, let me write it X as well. So X will be equal to your 3.3 into 10 to the power minus 5. So from here you can calculate the pH. How much is it coming? 9.26. I'm sorry, 9.52. Right. Fine. I guess you can try this and get an answer to this. Fine. Let's move to the next one. I think we have done this. Okay, let's skip this. Okay, this is a match the following. Okay, so column A has acid, base, conjugate acid, and a buffer solution, and there are definitions, explanations to these terms. So just check which of the following option matches it. So A, B, C, D, E. Yeah, absolutely. So A is acid, a substance that acts as a proton donor. We have base, a substance that acts as a proton acceptor. Buffer, mixture of weak acid and or base with its salt. Conjugate acid is a substance with one less proton than an acid. And you have the conjugate base, a substance with one proton more than the acid. Fantastic, right? Okay. Yes, you can try this. Which of the following is true? Like A is more accurate or B is more precise, and this we have done a lot of it. So just another one example. Just give it a try. So to check which one is more precise, you have to calculate the standard deviation, and to check which is more accurate, you have to calculate the mass. Sorry, the mean. Okay. So let me let me write the for A. You have mean as how much mean you are getting. It's like so it's 7.39. Something like that, and you're getting an SD of SD. On the other hand, for B, the mean. Okay, so the known pH is 7.4, which I've just missed to write it. So the value which we are which is known is 7.4. Uh, so A is any answer? Yeah, you can check from here. A is more accurate and B is more precise. Okay, from the mean and the standard deviation you can just check, but you have to calculate the mean and the standard deviation if this type of questions are coming. Okay, now the next one is, which of the following statement is or are true? Tap water is not used in biological experiments. A thermal shaker can control temperature and rotation speed. 100% ethanol is used for surface sterilization. Cooling centrifuge can be used to grow bacteria. A and B. Any other? Right. Fine. Only A and B are correct. Can you tell me what is the percentage of ethanol that we use for surface sterilization? Fantastic. 70. Great. Yeah. Good. So that's all. So here are the list of a few books from which you can take. Every one of you must have heard about it. Leninger, we have the Void, the Styrer. You can read these books to get a better idea about this particular lecture. Means this you can consult this book for all the lectures. Or actually, these three books are very important. It will clear all the basics of your on the biochemistry thing. Okay, so right then, so that is what I have for this week and for this live session. If you have questions, I can answer it, and I'm just dropping my mail ID. If you want to drop any email and you have any queries later on, just let me know. I will be very happy to answer you. Okay, how to calculate the standard deviation and how do I show it now? Okay, so you have to calculate first the mean, then you have to subtract each value of it. Uh, so you have to just type over here standard deviation and you can get. But first you have to calculate the mean value, okay, and then you have each of the data points. So you subtract it, you take the, how do I tell? Just give me a minute, I will just tell you. you got it. Yeah. So first, what you first do is basically you first find the mean. Okay, it's first as simple as that. Then it's the number of points that are there. So for example, you have 5 points. So like you have over here, you have 5 points over here, and the mean is 7.39. Okay. So for each of this, you have to subtract the mean. Okay. So 7.39 minus 7.32, 7.35 minus 7.39. Okay. And for each of that, you have to take the, you have to square it. Yeah. You have to take the square of it. Okay. Then you do it the square, and then you have to take the square root divided by the number of points that you are taking. So it's like mean minus each data point, the square of it, and divided by the number of data points that you have. For example, you have 5 over here, and the entire thing goes under the square root. You can just check it in Google, the equation. Okay, any more questions? Anyone else? Yeah, the NPTEL team will be sharing with you the lecture as well as the slide. So they will share it directly. I will upload it to them. They will directly send it to you. Yeah, thank you. Then we will again meet in the next week with a different lecture. And just one more thing, just if you have any questions, don't worry, just drop me a mail. Okay. Because I guess there are a lot of things that may not be possible to cover in a 2-hour lecture. So just drop it. Don't think twice. Just drop a text, because you can have questions when we are studying. Just drop it. Yeah, it's very important. With even with the questions of the assignments as well, right? So thank you. We will again meet in the next week, same time 6 p.m. Okay. Yeah, I think there is a class tomorrow, but I will not be taking it. There will be some another person who might be taking it. But yes, there is a class tomorrow. You people will probably get a mail on that or something. Okay, good then. Bye bye.